I'm really excited to be here, and it's been great hearing about all these different talks for the last couple of days. And so today I'll be telling you about some work that we've been doing on cancer metastasis. I should state that I'm more a biologist by training, but I'm really eager to kind of bring up a lot of open questions in this area and, really open to collaborations with all the data that we have. So my lab focuses on breast cancer, a particular subtype of breast cancer called triple-negative breast cancer. And this cancer is one of the more aggressive breast cancers. Up to about 50% of patients will ultimately metastasize to these main four organs: brain, bone, lung, and liver. And once patients have metastatic disease, survival is really poor. And really, but when we look at metastasis overall, the outcomes have not really improved. And so we can see that patients here with localized disease for a range of cancers, over the last, 10, 20 years, there's good survival for these patients, but there hasn't been much change. For the patients with metastatic disease, survival is, quite poor. And again, there's also been no change. So I just wanted to go over a few facts about metastasis that are important in terms of kind of how we approach studying it. And the first is that it's a very inefficient process. So it's estimated that, cells, a single cell has a really low probability of making it to form a successful metastasis, to the order of 10 to the -8. So lots of cells die. This is, a really hard process for cells. And so, metastasis and actually all parts of tumor growth require cell migration. And so there's a lot of modeling studies that show that even just for tumors to grow, if you have a tumor that only grows versus the tumor next to it, which grows and migrates, you get a much bigger tumor. So migration can also just support tumor growth. Then cells need to locally invade their surrounding tissues to reach either the vascular or lymphatics to be able to disseminate. And that process of local invasion, is dependent on motility. And then even for metastatic outgrowth, once cells have reached organs, there's really nice intravital imaging work that shows that once cells get to, this is in the liver, for example, these cells are constantly still moving, and they need to be able to move to be able to seed, again, these bigger metastases. And then if you inhibit cell migration, shown here, you get a lot less metastases in the secondary sites. As we've discussed yesterday, histology and kind of just basic H&E staining is still kind of the most commonly used approaches in the clinical space. But from these slides, you can't tell, which cell is migrating from these slides, right? There's very little information. In terms of what drives metastasis, there's been a lot of studies to, understand tumor cell intrinsic pathways that promote migration. And so there's been a lot of work looking at gene expression to try to identify signatures in cells. For example, here, list of genes associated with lung metastasis or bone metastasis. But, there's not a single really mutation that is associated with metastasis, which for, many cancer biologists can be very frustrating because there's such a focus on mutations. And also it's likely that many different pathways are contributing to this. And then we also have kind of the local environment that also contributes to migration of metastasis. And so a tumor that starts in the breast will see a certain environment. When it metastasizes to other organs, it will see a very different environment. And we have this whole seed versus soil hypothesis that was stated many years ago, that it's not just about the cell's ability to metastasize, but also their ability to respond or take advantage and survive in a foreign environment. And there are many components, such as the extracellular matrix, again, the lymphatics or vasculature, range of different stromal and immune cells, as well as nerves that are now, active components of the tumor microenvironment that contribute to these processes. And so there's also some clinical challenges that are important to keep in mind when it comes to treating metastasis. And so initially it was thought that metastasis happened quite late in the tumor progression kind of process, that you needed a tumor to be a certain size for there to be metastases, but that's not true. And so in multiple cancers, people have shown that they can detect disseminated tumor cells, both in mouse models and in human patients, quite early on in cancer progression. And so this makes it kind of, the idea that we can maybe prevent metastasis is much, harder to look at clinically because it's likely already started to happen. But I still think, there's a lot of value in studying migration and metastasis, because we want to be able to really predict which patients are going to be more metastatic. This is especially true in the context of breast cancer, where we're really good at diagnosing it really early now with mammograms and a lot increased access to that. And so when you have a patient that has a small breast tumor, do you treat it really aggressively or not? Being able to develop tests that can let you understand, like, is this tumor likely to disseminate or not, can help decide how much treatment patients undergo. These chemotherapy treatments, which are still, the main standard for many diseases, including triple negative breast cancer, are really aggressive with lots of side effects. We don't need patients to go through that if they don't have to. And, while some dissemination may have happened early, it's still, the more we can reduce dissemination, the lower the chance that metastases will actually form. So, but this is important in terms of kind of framing how we approach this problem, especially to reviewers. Another thing is that metastasis does not correlate with tumor size. So it's not that you have a bigger tumor that means you're definitely going to have bigger or more metastases. And so here are some studies where they looked at disseminated tumor cells in the bone marrow of patients with breast cancer. And, even, and they kind of classified the tumors by size. And, see, there's no difference in the amount of cells that have disseminated early from these tumors. So, the size is not impacting the dissemination. And then when you look more kind of large scale studies, the relationship between tumor size and then either presence in the lymph node, which is one of the first steps of dissemination for breast cancer, or looking at overall survival, which, is linked to metastasis because that's what patients die from, there isn't a linear correlation. And there's a bit of a relationship initially, but then it really kind of flattens out. And so, this is challenging because in terms of drug development and how clinically response is assessed in patients, it's assessed by this RECIST criteria, which is basically, how big a tumor is. So clinically, there's just, so much focus on the size, but that doesn't inform a lot necessarily on the amount of metastasis that has happened. And so, that's still something that, needs to be reconciled in terms of, how we can treat metastatic disease. So today I'll kind of focus on 3 different areas. One is, how best can we study metastasis in the lab? How do we integrate the microenvironment? And then, how does this affect these different processes affect response to therapy? So what assays are the best to study metastasis? Now, I review lots of grants for the NCI, and I'm always surprised that so many people still use this Transwell assay. This is like what is really, really commonly used for many people, where you have this membrane and you place cells on top and they migrate through this porous filter. This is migration through a membrane. There's not really that's not representative of an actual matrix. It's really hard to do in 3D because some people, have add a bit of layers, but it's not quite the same, and you can't image your cells live moving, right? And but this is still really, really commonly used. Similarly, the scratch wound assay is also really commonly used. I see lots of grants with this, but that's really hard to do in 3D. It's confounded by cell proliferation. And, it models cells migrating collectively, which is important for some aspects of dissemination, but not all. And so these are still the most, common assays used, even though we've come such a long way in terms of the, imaging and assays that we can do. So in order to see which assays that we use are the best and most predictive of metastasis in vivo, we took the kind of 6 most common human triple negative breast cancer cell lines that are used in the literature. We heard about some of these yesterday. And these come from a range of different patient backgrounds. Some are mesenchymal, some are more epithelial, different mutations. And we compared their behaviors and tried to figure out which assays were more predictive or correlative of their in vivo behaviors. Because it's really hard to know which assay is the best to be able to model metastasis. We don't want to waste our time running lots of different assays if we can just find one that works better. So, these tumors, we injected them all in the same way, same amount of cells, same environment to keep this, really the same. You can see that in terms of tumor growth, there's a lot of variation between how they grow. We compared metastases to lungs and liver because those are the most common organs that cells metastasize to in mice. And so in terms of the lung, you can see that we have kind of 3 cell lines that emerge as highly metastatic versus 3 cell lines that are more poorly, that don't really metastasize at all. But if you look at these 3 cell lines, there are differences in terms of the growth. So again, there's not this necessarily like direct correlation between growth and metastasis. In the liver, we get even something different where the 3 kind of higher metastasized cell lines do metastasize, but then we have this other BT20 cell line, which didn't go to the lungs at all, but it actually went to the liver. So again, kind of the environment in different organs seems to matter for these different cell lines. So I'll just run quickly through the different assays that we did, but we looked at cell shape, as been discussed, with a range of different parameters, and you can see that these cells have quite different morphology when we see them in the dish. We looked at phenotypes in 2D because these are, generally much easier to study, both in terms of their migration speed, and we looked at both speed and persistence as well. So, how directly they move on a specific path. And we also looked at their viability. And so these cells kind of all have, pretty similar viability, but can have much more drastic changes in cell migration in 2D. And in 3D, we looked both at single cell behaviors where we seeded cells in collagen gels, and collagen is the most abundant matrix protein present in the breast tissue, collagen 1. So just trying to keep a simple kind of 3D environment. And we saw, really different behaviors in terms of their invasion speed as well as their persistence. And we also use a spheroid model, which is commonly used where, you seed cells in a clump together and the cells will invade out. The spheroid will also grow. So this assay does take into account both invasion and proliferation, and saw differences there. And so we just did some very basic modeling to be able to figure out which assays, could explain the variance that we found between these different cell lines, between our poorly metastatic ones, our highly metastatic ones, and our more intermediate phenotypes. And what we found was that actually 3D persistence and actually decreased 3D persistence was associated with higher metastasis. So it seems like the cells that, were kind of moving more randomly seem to be more metastatic. And we also found that cells, their ability to make protrusions and to kind of change from more round cells versus protrusive was also really important. And so, now we've tried to focus on using kind of these assays. Looking at cell shape is obviously very easy, and you can do on a large scale to be able to ask these questions for lots of different cues that you might be looking at. 3D assays obviously require more live imaging capabilities, but can also be very informative. There have been other assays that have been other approaches that people have used to try, and this is mostly in breast cancer. We heard also about the Dexter's lab work in other cancers and others. So I'm just focusing on some recent breast cancer assays here of trying to use more complex assays to try and predict metastatic potential. So this is one where they have this channel and the ability of cells to kind of migrate through a really thin microfluidic device, which kind of mimics their ability to migrate through a tissue environment or kind of vasculature, and showing that that's correlative with metastatic potential in vivo. And then recent work from the Engler lab where they look at how the adhesion strength, so not looking at the shape, but how strongly these cells adhere where they seed them and then they shake them and see how many come off, and so how strongly they're attached. And that also is associated with metastatic progression. So, these assays are more challenging. I think, we are happy that at least the cell shape is very straightforward and amenable for more people in the community to be able to do. So again, we find that cell shape and 3D persistence are the most predictive factors. And again, we show that, these tumor models do mimic this fact that we don't have this perfect correlation between growth and metastasis. And that's important in terms of kind of studying progression and how different drugs or genetic interventions, might impact effects on growth versus effects on metastasis. I see so often that many people assume that because something is going to have an effect on growth, it's going to have an effect on metastasis. And, that's just not the truth. And so it's important to look at these processes separately and, understand behaviors on both of these phenotypes at the same time. So, here we've been looking at kind of tumor cell intrinsic properties and, what about the tumor microenvironment? And I add a little bit of Mina to make you think of what I worked on during my postdoc just to introduce this. So my postdoc, I worked on a cytoskeletal protein called Mina, different isoforms that, and in vitro, when you express these different isoforms in cells, you see very little difference in the migration speed for these cells. When they form tumors in vivo, the tumor size is the same, but you get this huge difference in metastatic potential, where these cells that express this more aggressive isoform here in green, metastasize a lot more. And so, again, in vitro, you see no difference here. So why, do we have this big difference? And that's because the processes involved in this pathway are sensing changes in the environment. And so, there are many cues within the environment that can drive migration. And today I will focus mostly on the extracellular matrix. So in the last 10 years, there's been huge progress in understanding the complexity of the extracellular matrix, thanks to proteomic studies that have really delved into kind of characterizing the breadth of the proteins that are present in these tissues. I think there's a lot of focus on collagen 1, obviously, is really abundant, fibronectin, but there are so many other matrix proteins that are present in our tissues that play important roles. So the composition of the extracellular matrix, matters a lot. There's many changes in the matrix that can contribute to cancer progression, and these are summarized here. Changes in stiffness, changes in the cross-linking and structure of the individual fibers, how these retain different molecules. So there's a lot of different ways, but today I'll focus on kind of the composition of the matrix. And so when we study the matrix, when we look, in vitro, we can, buy a recombinant protein. You can make them if you want, but they're pretty challenging because they have a lot of post-translational modifications. But we can buy, recombinant protein, and, many talks were talking about these and coding them, and you add your cells on them. And these are great because, you can add a specific cue and look at the specific response, and you can dissect the mechanism by which a particular matrix protein affects, a particular phenotype. And so we've been really trying to dissect, now that we have these kind of proteomics studies, with 150 matrix proteins present in a breast tissue, what are some of the other matrix proteins that, are contributing to the metastatic phenotype? And so we've leveraged, publicly available proteomics data sets and some of our own. And, we've been trying to use our cell shape assays to try to screen a lot of different matrix proteins to identify, matrix proteins that are contributing to metastatic phenotypes. And so, just as a start, we looked at kind of the first 4 that are the most common. So collagen 1 and fibronectin, which are obviously well studied. A couple others, collagen 4, present in the basement membrane, tenascin C, which is, found in that lung metastatic signature. And so we can see that, these individual proteins have drastic effects on cell shape, and therefore, drastic effects on cell motility and progression. And so we screened, quite a few different matrix proteins, and we landed on collagen 6, which is, another family, another member of the collagen family that's, plays a lot of role in joint integrity and function, but is also really abundant in breast tumor tissue. It induces really drastic differences in cell shape, and we validated in our different 3D assays that, they do also induce invasion, in these, in different models. So it's been really helpful to use these, again, to identify novel ECM drivers. So another aspect of the matrix is that it's secreted by lots of different cells. While fibroblasts are most famous, known for their role in secreting matrix, they're not the only ones. But there's lots of work that can be done on, seeding cells in a dish, letting them secrete matrix, and then remove them and then add other cells on top that you're interested in. And so this is great because you get a more diverse matrix with multiple proteins and some cell remodeling, but it's still only generated by one cell type. And so we have, lots of work going on to try and dissect which are the main contributors. And again, fibroblasts make these beautiful aligned matrices. They're really great. Cells migrate on. But we've discovered that other cells also secrete matrix, like adipocytes are in the breast tissue, and they secrete a ton of collagen 6, and that can contribute to cancer progression. And more recently, we've been studying the role of nerves, and nerves as well secrete a ton of matrix as well in the breast tumor tissue. And that makes the cells migrate a lot more when you have matrix generated from nerves. And so, trying to understand kind of, what all the different contributions of these cells are to the matrix that's in the tissue is quite challenging. And so these are things that we are looking into. But lastly, we have to remember that even if in vitro we look at one matrix Q, in the tissue, there's a lot of matrix proteins that are present. And so how do you go and look at kind of the whole tissue effects of the matrix? And so you can take a tissue and decellularize it with detergents to obtain a matrix scaffold. But this has been commonly used in terms of tissue engineering, but many people dissect this up and kind of break up the matrix, and you lose so much information by doing that, even though this can be used for regenerative approaches. And so we kind of repurposed this, and we generated tumors from mice using different genetic mouse models in breast cancer or injected cells. And we isolated the mammary glands from healthy mammary glands and then tumor-bearing mammary glands. And we decellularized, and we, and so this is what it looks like when you have your tumor tissue, and then you decellularize it for a couple of days, and you get this nice matrix sponge scaffold, which has all the matrix that's in the tissue, no matter what cell secreted it. And then we seed cells on top of it, and we look at their effect. And so here, what we were able to show is that, if you seed cells on a native kind of mammary gland matrix scaffold, there's not as much migration. But when you seed cells on a complex matrix generated and isolated from a tumor, suddenly you have a lot more migration, and you also have actually a lot more proliferation. And so, this is like the first demonstration that the whole tissue matrix of a tumor is pro-proliferative, pro-invasive. And so that, you need to keep in mind kind of the whole, matrix that's present in the tumor tissue. But then, how do you go and figure out, well, which ones are actually driving some of these effects? And so here what we did is we knew in these tissues there was a lot of, collagen 6, which I'd mentioned. And we did a lot of work in vitro to dissect the mechanism by which collagen 6 drives invasion. And so I won't show all the details, but we found that it signals through this NG2 proteoglycan receptor and crosstalk with EGFR and MAP kinase signaling to drive invasion. So then what we did is we went back to our decellularized tissues with the whole tumor matrix, and we reseeded cells in the presence of an NG2 inhibitor. And so we could block this pathway that we know is activated by collagen 6 and see if that affects migration. And we found that, in this tumor model, when, you have this increased migration, when the cells are seated on this tumor ECM compared to the mammary gland ECM, and, when you have an NG2 receptor blocker, you reduce that. So this allows us to still ask, what pathways are important in driving these effects within the whole context of the tissue, and whether these, matrix proteins we identify, have effects on their own when we have them as recombinant proteins, also, are driving effects in the whole tissue tumor ECM context. The other thing to remember with matrix, and this is true with other cues, is that, these cues are not necessarily homogeneously distributed within tumors and within tissues generally. And so I did a lot of work in my postdoc on studying kind of gradients of matrix proteins. And so I worked on this protein called fibronectin, which, famous matrix protein. And when you look at how fibronectin is distributed within a tumor, you can see that in the normal breast, we don't see as much. But in the tumor, you see this accumulation of fibronectin around these blood vessels, and then these kind of lower regions of lower fibronectin inside the tumor. And so we did a lot of work to look at how, a gradient of a matrix protein, does that impact cells as well? And does that have an effect in terms of maybe in this context of fibronectin attracting cells, to the blood vessels in terms of this? And the great responses to gradients of matrix proteins are called haptotoxis. And so, spoiler, we found that, fibronectin gradients do kind of attract tumor cells. And, to look at this, we developed microfluidic devices where we could develop gradients. And so these are 3-chambered devices where you then use a pump to flow in a matrix, and you then push it through, and it goes through these microchannels in order to deposit as a gradient on the middle channel. And so we can quantify gradients. We can do this in 2D or in 3D with cells in different hydrogels, and we can get these really nice gradients of matrix in these channels. And then we do live imaging, and we not only look at the speed of the cells, but also their directionality. And so we quantify this forward migration index, and when cells are moving towards, your cue of interest, they have a positive forward migration index, and when, they have a negative one, it means that they're repulsed. And so once we identified fibronectin around blood vessels, we were also looking at collagen 6. And, we could see that there was a lot of collagen 6 like around the edge of the tumor here. This is because a lot of it, again, is secreted by adipocytes, fat cells that are present in the healthy breast and that surround these tumors. And so we saw these really nice gradients of of collagen 6 all around the edge of these tumors. So we looked at, whether collagen 6 could also drive directional migration. And so we made gradients of collagen 6. And you can see that, here we have, increased migration. So we have more speed, which is what we knew from this. But we also have increased directionality towards the collagen 6. And if you look at kind of the shape of the cells along the gradient, you can see that kind of as, the gradient gets stronger and you have more collagen 6, you get kind of more changes in the morphology of the cells. And so, this, we're still kind of investigating how this all fits in with, in the context of metastasis. But, it does bring up this concept of looking at kind of homogeneous cues that drive migration or drive effects versus directional cues that drive, different migration processes within tissues. And again, it's not likely, unique to tumors by any means. So, we've shown that the matrix is a major driver of tumor cell invasion and progression, but, can we leverage that and identify properties of the matrix to try and predict more invasive tumors within breast tissue, for example, and predict metastatic potential? Those are things that we're really interested in. And then how do we kind of integrate these cell intrinsic properties of some tumor cells being more metastatic than others? And then, how they then respond to cues within the microenvironment to, allow them to progress or not. And so those are kind of questions of, how do these cells compete or manage these competing inputs? And then, this is not the only cue or gradient that can drive migration. Changes in pH, soluble cues, changes in stiffness, electrical properties, oxygen levels, all of these things are changing in tumors. All of these have been shown to contribute to migration. And a lot of these can also occur in gradients within tumors. So, how does a cell, integrate the matrix cue, but the other cues as well? Like, how does it face these different competing cues? Which one takes over? Does it depend on the cell? These are kind of really important questions that we want to try and answer. And, how do you bring together this kind of random migration of the intrinsic cells' ability to be able to move around versus them responding to different cues in the environment? So these are some open questions. And then lastly, I'll just share small insights in terms of, how does this then affect treatment and how, how patients respond to certain drugs. So chemotherapy still remains the standard of care for this triple negative breast cancer subtype. Immunotherapy has been approved, but has limited effects in the population. And so again, you get a patient that comes in, they will get a diagnosis, they will get some neoadjuvant treatment, so different chemotherapy drugs. But many of them, they still have a tumor after that. So then they'll undergo surgery. You hopefully get everything out. But then you have, about up to 50% of patients that their cancer comes back. And so, what we're trying to, understand is, why does chemotherapy not kind of reduce this metastatic recurrence? And why is chemotherapy not kind of having a stronger effect here? And does it have anything to do with, migration, metastasis? So when we look at the feature of resistant tumors, it's also for you, Gaudens, these tumors that are resistant have high migration gene signatures. So there's a lot of migration that's associated. There's also a lot of matrix remodeling genes and proteins that are found in chemo-resistant tumors. And there's a lot of metabolic dysregulation as well. Those are kind of some of the three main kind of categories of pathways and genes that are associated with chemo-resistant tumors. And, all of these things are linked, but I'll just share a little bit of data on some of these. So, metastatic cells can drive resistance to chemotherapy. So again, this some work I had done in my postdoc where, you're comparing these three cells. The green ones have more metastasis than the control ones. And so you can see that even a cell being more metastatic, it affects how it responds to a particular drug. This is paclitaxel, which is, one of the standard drugs used in breast cancer. So these cells will survive more in response to paclitaxel. And so when you treat mice with these tumors, the control mice, their tumor slows their growth. So that's, it works. We know this is a dose that works, but in tumors that are highly metastatic, there's no effect here. And so, there can be, if you have a whole population that's highly metastatic, then, there is resistance to chemotherapy. But usually these tumors are heterogeneous and there's a range of population that's present. So we've been trying to go back and look at, okay, across these cell lines, if we look at cell shape, do these drugs have different effects on cell shapes, right? So we use the 4 kind of standard drugs that are used interchangeably in different patient triple negative breast cancer. And we identify doses where they all induce about 10 to 20% cell death. So there's some cell death, but there's still a lot of cells that are alive. And you can see that the effects on cell shape, and I've just shown here kind of how protrusive a cell is, can be very different. So doxorubicin, decreases, makes the cell more protrusive, versus paclitaxel, makes the cells less, more round. So each of these drugs, even though they're all killing cells, kind of the same level, same effect on cell growth, very different effects on cell shape. And then, if we look in vivo, so we treat tumors, we picked these 2 drugs to start with, Taxol and doxorubicin. These, have different mechanisms of action, how they act on cells, but, we have doses where they shrink the tumors. So we know they're killing cells. This is happening. One thing we haven't discussed is what about migration live inside the tumors? And so I've done a lot to integrate intravital imaging as well into some of our work, where, we can take mice that have tumors, we dissect them, open them out, and put them flat down on a 2-photon microscope under anesthesia. The tumor cells are fluorescently labeled, and, we can see cells that are moving within these tissues. And so if we look at, these mice where the tumors have shrunk, how is migration in vivo? Well, actually, like Taxol does decrease migration, but doxorubicin does not. It does nothing to migration. So again, kind of the effects on the size and the growth, are the same, but the effects on migration live inside a tumor are different. So this is something that we are investigating further. And then lastly, we also wanted to look at effects on the matrix. Maybe it's not just that, again, because chemoresistance was associated with changes in matrix in patients. And so we have a great assay to be able to look at the matrix after chemotherapy treatment. And so we treated mice with breast tumors with, again, these 2 different chemotherapy drugs. And, both of these drugs, again, slow down tumor growth. So we're seeing an effect on growth. This would be, quantified as a response within a patient. But there's no effect on lung metastasis, on dissemination. So, we're kind of replicating this phenotype of, tumors that are resistant. And so we, at sacrifice, we got these mice, we took out the matrix from these mice, and that was made after chemotherapy treatment, and we reseeded fresh, naive cells on these. And what we found was that on matrix that was from chemotherapy-treated tumors, either paclitaxel or doxorubicin, we saw a lot more invasion. And so this shows that chemotherapy is also changing the matrix and making it, more attractive for migration for these cells. So, in the context of these chemotherapy treatments, you have effects on cell proliferation that on tumor size, and these are shrinking. But, you may have different effects on cell migration that's happening in the tumors. You also have changes that are making the matrix more attractive. And so, how do we kind of reconcile all of this, and how can we use this in order to, better understand what patients might be, where this might be happening, that might be a higher chance for recurrence versus a patient where it's not happening, and we can kind of, treat them less aggressively and just kind of watch and wait. So that's kind of where we're heading towards this. And again, kind of how we can integrate both, changes in terms of like the tumor cell intrinsic properties of tumor cells, but then also kind of the cell extrinsic drivers of migration. Again, we've been focusing mostly on the extracellular matrix in this context, and how do these competing forces kind of work together. So hopefully it's giving you kind of food for thought in terms of, how we look at migration and metastasis and different questions that are open. And so, I think the members of my lab, past and present, who did a lot of this work, and our funding sources, and I'm happy to take any questions.
And we have time for questions.
I'm really good talk. I learned a lot. I really like your study and to figure out what are the cues that from the microenvironment that cells, the cancer cells, trying to interpret. My question is, how different are the experiments set up from different labs that make it possible for people to interpret this data from different labs, trying to figure out and compare those results?
Yeah, so I think the first biggest issue is that we work with cancer cell lines, and these mutate a lot. They alter their chromosome, makeup a lot. And so, as Gaudens mentioned yesterday too, when you grow these cells for a long time, they can change a lot. And so just, having cells from different labs, the same cell line from different labs, can behave differently. So this is a huge problem in cancer generally that makes it hard to integrate data, even in the first place. But, I think in terms of, many of the labs have different assays. So I think those still could be integrated together. In terms of, looking at different properties or different assays to bring them together. But, I think that's a good point, and something that we need to kind of make sure that, investigate that more and make sure how we can try and make it so all this data can be kind of usable all in the same way. So, yeah.
Okay, very nice. It's an intriguing result that Taxol changes the matrix. So if you have an idea, is that fibroblast in the tumor which does it, or can you constitute that in vitro?
Yeah, so we have a whole paper hopefully coming out really soon on that. In vitro, looking at the fibroblasts, we were able to really replicate the results in terms of activation of alpha SMA. So the fibroblasts become kind of more activated, changes in cell shape, produce more matrix. That matrix is more invasive. It's been more consistent in vitro with doxorubicin than Taxol. Taxol, the effects are milder, even though in vivo we kind of saw similar effects, although slightly stronger with doxorubicin. So we have really strong phenotypes with doxorubicin. And it seems to be through increased TGF-beta secretion and signaling via receptor tyrosine kinases and downstream SMAD action. So we've kind of pieced that out. So there's definitely, some fibroblast effect. But yeah, likely seems to be in our case more doxorubicin driven, but so yeah.
Take a few more questions.
And maybe I missed it, but what is the pathway that regulates the collagen 6 production?
Yeah, so we have not fully figured that out. What we have found is also that collagen 6 is really high in the mammary gland of obese individuals, and that obesity is associated with breast cancer progression. So we see kind of increased matrix production by adipocytes, but we haven't figured out exactly kind of how that happens. It does kind of go up over time as the animals gain weight, and we can see more production there, but we haven't kind of pieced out specifically what is driving that.
behind, yeah.
Yeah, yeah. So yeah, that's a very interesting talk. At some point, you mentioned that like the decreased 3D persistence that sort of promotes like metastasis. So I was just wondering like how that happens.
Yeah. I mean, we saw that was associated with more metastasis, so cell lines that had decreased persistence. I mean, I assume that it's that, you know, if they're still migrating, like they're just kind of migrating like crazy. And again, because it's such an inefficient process and it's so hard for these cells to metastasize that the more they move, the more they can just try out the environment, find things that work, disseminate, the higher chance there's going to be that are more that grow, that get out and that can grow in other places. So I think it just gives them more ability to interact with the environment, more chances to kind of forge a path in order to, you know, they're not moving as kind of just in one direction. They're sampling things a lot more, and that makes them more able to, you know, successfully then spread to the next step. The more cells get out, the more cells will likely, you know, make it to the end.
We have to cut off questions there, but there is a coffee break later. Let's thank Madeline again.